For any assignment related queries, call us at queries, call us at +1 678 648 4277 1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Question 1. Bromodomains are protein domains that bind to acetylated lysines, similar to SH2-phosphotyrosine interactions. J. Bradner at Harvard has recently developed JO1, a cell-permeable small molecule, mass 642 grams per mole, that binds and inhibits a subset of bromodomains. In the lab, you would like to determine the affinity between JO1 and BRDX, a mass 24,354 grams per mole, your bromodomain of choice. A. Design an SPR experiment to quantify the interaction affinity between 1Q1 and BRDX, including proper controls and which molecule is immobilized. The details of exactly how SPR works are not critical to answer this question. Justify your immobilization selection by calculating the number of captured molecules needed to obtain a 10 RU signal change in both configurations. Note: assume a 10 mm 2-chip surface area. To maximize the signal obtained with each protein binding event, we want to immobilize the lighter protein, JO1, on the surface. During the SPR experiment, we then flow solution containing the larger protein over the plate and measure the RU versus time profile. One important control is to run solution containing the larger protein over a plate that doesn't have any immobilized protein. Changes in RU during this control may indicate potential nonspecific binding. Nursingassignmenthelp.com. Immobilize BRDX, immobilize. Clearly, by immobilizing JQ1, we need far fewer binding events to obtain a 10 RU shift, and thus it is the logical choice. B. Given the following parameters, K is equal to 3 times 10^4 mol 1s-1. Another important control is to run the experiment multiple times using different ligand concentrations. The results between these experiments should be consistent. Con equals 3x 10^4 mol 1s minus 1 cof equals 4x 10-2s minus 1. Nursingassignmenthelp.com. How long will it take to complete the experiment using a ligand concentration of 200x 10-9m, assuming the association reaches 90% of its equilibrium value before ligand stops and 90% of the associated ligand dissociates by the end of the experiment? The time required is composed of the association time and the dissociation time, for the association phase. For the dissociation phase. Nursingassignmenthelp.com. Note that corresponds to the value of RU after the association phase is terminated, not the actual equilibrium RU. Understanding the derivation for was vital for understanding this point. Therefore, the total time is C. Although SPR is a very powerful approach, it does have some limitations. Provide 2 limitations and describe how you would know that they are affecting your measurements. 1. Must immobilize ligand on surface. Attachment can affect chemistry, such as blocking the binding site. You would know because you would not observe any binding even when you would expect to. 2. KD must be between 10^4 and 10^12 m. You would know because below 10^12 you would see nearly instantaneous binding and no dissociation. Above 10^4 you would never get any binding. 3. Non-specific binding can lead to spurious results. You could see unreasonable values of KD. You could run a control with the ligand solution before immobilizing the protein on the surface to see if any binding occurs. This binding would then indicate potentially spurious binding. Nursingassignmenthelp.com. Question 2. Overexpression or mutation of the epidermal growth factor receptor, EGFR, has been associated with a broad range of cancers. As discussed, there are many companies that have developed molecularly targeted therapeutics, and several of these compounds are ATP analogs designed to inhibit EGFR. One of these compounds, gefitinib, is currently approved for lung cancer patients with high EGFR expression or with selected activating mutations in EGFR. Km for the EGFR ATP interaction is 50x10-6m, Cat for EGFR ATP is 106 SEC1, and K for the EGFR gefitinib interaction is 22 × 10⁻⁹ m. ATP concentration in the cell is 3 × 10⁻³ m. Due to dose-limiting toxicity, the intracellular concentration of gefitinib is limited to 100 × 10⁻⁹ m. How effective is this dose at shutting down EGFR activity? We can use the regular equation and the equation with a competitive inhibitor to determine the reduction in reaction rate as follows. NursingAssignmentHelp.com. This means that the rate of the reaction with the inhibitor is 93% that of the reaction without the inhibitor. Percent inhibition is given by 1 to 0.93. Therefore, it only reduces the rate by roughly 6.94%. NursingAssignmentHelp.com. B. How much does EGFR inhibition change in cells expressing 106 copies of EGFR versus cells expressing 104 copies of EGFR? The equation for V/V is not dependent on the enzyme concentration, so the percent inhibition is the same for both 104 and 106. However, the total number of inhibited receptors is different. 104, 700 receptors inhibited 106, 70,000 receptors inhibited, 100-fold change. C. Why is a strong increase in expression of EGFR often associated with cancer, even in the absence of ligand stimulation? The increase in the number of proteins allows the proteins in the cell to sample more conformations. Thus, more EGFR receptors spontaneously open their active site and allow ATP to enter even in the absence of the activating ligand. As a result, these cells are more prone to dimerizing and activating the EGFR pathway and its associated anti-apoptotic and pro-survival prognosis. Other answers accepted included references to EGFR activating mutations, altered role of feedback in cancer such that more and more receptor is generated, etc. Stating that EGFR drives proliferation which runs rampant in cancer is not enough here. D. Describe how the mechanism of action of a type 2 inhibitor differs from that of an ATP analog. An ATP analog binds competitively to the ATP binding pocket, blocking activation of EGFR by preventing interaction with its necessary cofactor, namely ATP. A type 2 inhibitor binds the DFG out motif when the enzyme is in the inactive state and blocks the opening of the activation loop, which is necessary for the enzyme to bind ATP and substrate. With respect to ATP, type 2 inhibitors can be considered a non-competitive inhibitor, although it is more complicated than this in reality. NursingAssignmentHelp.com. E. Given a cancer cell line with an activating EGFR mutation, would you use a type 2 inhibitor or an ATP analog inhibitor and why? Due to the mutation, the kinase will always be active. As a result, a type 2 inhibitor will be completely ineffective as its function is to prevent the kinase from transitioning into the active conformation. As a result, the ATP analog would be the correct treatment option. NursingAssignmentHelp.com.